So Dr. Vineet Ahuja is professor from Department of Gastroenterology from AIIMS, New Delhi. He's done his MD DM from there, and he has been working on inflammatory bowel disease, abdominal tuberculosis, gut microbiome, GI immunology. He has got 30 book chapters and lot of publications with 62 H index and more than 20,000 citations. His papers are in NEJM and Lancet Gastroenterology. So also he was Associate Dean Research at AIIMS New Delhi. So with this, I invite him to speak on AMR and FMT.
Thank you very much, ma'am. Thank you for the kind introduction. I don't think it is in any gym. So I'm going to talk about antimicrobial resistance, but I thought it's a diverse audience out here. So I thought that I'll take you through some very basic terms, and that is what is MDRO, what is AMR gene, what is horizontal gene transfer, what is resistome, how are they important, why is colonization resistance very important. So if it comes to fecal microbial transplantation, comes a major question. Can FMT be a useful intervention in this kind of setting? Do we have definite data as yet or not? So AMR clearly is a problem which is not only for the developing nations but also for the developed world. It's clearly a problem of global concern, one. Secondly, mortality with AMR is rising, and by 2050 it is projected that the mortality through AMR would be maximum compared to any other disease, whether it is cardiovascular or whether it is cancer. Number three, AMR is something which is possibly also the link in bit for One Health. AMR develops when antibiotics are given to animals. AMR develops when antibiotics are given to human beings. So that completes the circle. So it's also a very, very important concept for One Health. And that is, I think, another concept which is very, Professor Ramachodi works a lot on that. And so how does AMR actually develop? Simplifying, harmful bacterial colony mutation creates an antibiotic resistant cell. Antibiotics create selective pressure, killing the non-resistant cells. That leads only to antibiotic resistant cells, which proliferate a lot. And not only they proliferate a lot, they spread it around. They spread it horizontally with the help of what we call as mobile genetic elements. Plasmids, which are circles of DNA, transposons, which are small pieces of DNA, or phages, viruses that attack germs and can carry DNA from bacteria to bacteria. Now that is important. Phages can transmit antibiotic resistance, and that is where the importance of gut microbiome also comes in. So these three mobile genetic elements, they can cause horizontal gene transfer with the help of transformation, conjugation, and transduction. Transduction is done by bacteriophages. Again, as I told you, that is an element of interest for us because bacteriophages are found in the gut microbiome, and that is again what we want to manipulate subsequently. What is MDR or multi-drug resistant organism? Simply said that they should be resistant to at least 3 of the 6 antimicrobial groups. And for XDRO, it is at least resistant to at least 5 of the 6 antimicrobial groups. Then there's a term known as antimicrobial resistance gene. What is it? It is a free DNA. This free DNA can get incorporated into the plasmid, and as a result, it can cause transmission of antimicrobial resistance. So it is a very important part of this concept. So MDRO is important, and MDRO can be things like vancomycin-resistant Enterococci BRE, carbapenems resistant Enterobacteriaceae CPE. Antibiotic resistance genes can be VANA, BLY, etc. So both of these are very important if you are planning a study on antimicrobial resistance. What is resistome? Resistome is what when we talk about microbiome. Microbiome is the total gene collection of all kinds of organisms which are there in the reservoir or in the ecological reservoir. Resistome is the collection of antibiotic resistance genes in a community of bacteria. It includes genes in both pathogenic as well as non-pathogenic bacteria. So as a gastroenterologist, how do I get into it? In a study which was done at our center, it was seen that in rural population, healthy population, when we looked at MDRO carriage, it was seen that 27% of individuals from rural northern India were carrying MDRO organisms, and the ESBL carriage was around 44%. So 27% of healthy rural people were carrying MDRO organisms. Whereas a study which we had done earlier, where we had looked at gut microbiome in people living in rural area, urban area, people living in Ladakh, considering that, okay, maybe they have a different kind of diet and all. It was seen that amongst these three healthy groups, the best microbiome was either in people living in rural area or people living in Ladakh. And here, another study, we saw that people living in rural area had 27% carriage of MDRO. Subsequently, with the Rohtak group, Dr. Krishan Kansharma, we looked at patients of ulcerative colitis who were living in rural areas, and it was seen that ulcerative colitis patients had even higher level of MDRO as compared to the healthy controls living out there, suggesting that not only MDRO is out there in the general population, it is more in the diseased population, and it also seemed to be related to the disease activity. So clearly, as a gastroenterologist, there was in a sense, there was some room for intervention out there. You know that gut microbiome is a new and essential organ. Microbiome manipulation therapies are the future of medicine. So comes in fecal microbiota transplantation, which is infusion of a fecal suspension from a healthy individual into the GI tract of an individual. Now, there is at present, whatever we may talk about, only one globally approved indication for FMT, and that is recurrent Clostridium difficile infection. Rest, any other indication, if it is there, is part of research trials. It is, but that indication is so much, it's like a volcano burst in 2013 when the NEJM paper came up. And obviously, you see a lot of people out there with a torchlight because people really wanted to work in every disease. And no doubt, there are a lot of trials which are coming up, and they encompass diseases not only from GI disorders, hepatic disorders, to schizophrenia, Parkinsonism, obesity, autism, any kind of disease you talk about, there would be a FMT trial going on. Clearly, it's a kind of movement which is building on. There's a recent study which came up which talked about poo milkshake, which boosts the microbiome of C-section babies, where fecal transplantation was given to children after cesarean section and was found to be promising. We had done a trial, FMT trial 2 years back, which looked at fecal microbial transplantation with anti-inflammatory diet for induction of remission and maintenance of remission in patients with ulcerative colitis. FMT was given for 8 weeks, followed by maintenance with anti-inflammatory diet, and the trial found significant, effective results in maintaining clinical remission and subsequently maintaining it for inducing clinical remission and maintaining it for a period of 1 year. And subsequently, we looked at the microbiome metabolite changes. So obviously, when it comes to AMR, our antennae were up. That we have here a condition where healthy microbiome is there, there could be because of colonization resistance. And whenever there's a disruption of the microbiome, the colonization resistance goes away. And subsequently, if you give fecal microbiota transplantation, is it possible that you'll restore the diversity? If you restore the diversity, you restore the colonization resistance, and obviously the patient becomes well. Is it possible? So obviously, before you embark on any such study, you would go and review the literature. The present literature on this is this. This is what I would call the footprints of FMT in AMR. So we find that if we can look into all studies of human studies, that is of microbiome manipulation, we find majority of them, or 99% of them, are case reports, cohort studies, or case series or something like that. There are only 2 small randomized control trials which are available till now, one with 44 patients, another one with only 11 patients. So one with 44 patients randomized into 1:1 arm, FMT versus no intervention. 41% response with FMT, 29% response with no intervention. There was a signal, but it was not significant. Second study was done in renal transplant patients. This may interest Professor Ashish Sharma, in the sense that it had only 11 patients. So they had a kind of protocol where they give observation first or FMT first. So 5 to 6 patients in each arm, and followed them up and found that ultimately 8 out of 9 patients, there was a response when it came to AMR. So this is all the data which is there. No blinded study, no placebo controlled study. So we thought of a study and have just completed it, and I'll take you through the results of this. This is efficacy and tolerability of FMT for microbiome MDR intestinal decolonization and AMR gene depletion in patients with GI diseases, a double-blind, randomized, sham-controlled trial. This was on the basis of an ICMR grant which we had received. And obviously, such a kind of study would require a good team. So we had a very good team. Dr. Himanshu Naring did all the clinical trial part, recruiting patients and doing FMTs, etc. Dr. Pura Mathur from AIMS, she did the stool culture and the AMR gene sequencing part. Dr. Bhavatosh Das from THSTI, he did all the microbiome, and the virome work out there. So we had a very good team which built up. The primary objective was to evaluate the role of FMT in causing AMR gene depletion and intestinal decolonization from MDROs. Secondary objective was to look at what happens to the phage profile changes pre and post FMT, and also to look at donor engraftment. It was a randomized double-blind sham control trial, patients who were in gastroenterology ward and ICU. And they were given randomized to one session of FMT, or the other arm, which was the control arm, was sigmoidoscopy with saline injection. Detection of MDR organisms in stool was done at 4 weeks. Sample size was around 70, which makes it probably one of the largest RCTs done on FMT for FMT and AMR at the present. And we also did a stratified randomization. We decided that when you talk of GI diseases, 30 patients of pancreatitis, 30 patients of cirrhotics who are admitted to ICU, 10 can be any miscellaneous GI diseases, and let us see what happens. So we had these patients who were in ICU or the ward, tested positive by rectal swab for AMR gene and culture, and subsequently they were randomized. After they were randomized to FMT versus the other arm where only saline injection was given, followed for 4 weeks, and subsequently again tested for the bacterial profiles. As I told you, the randomization was for pancreatic, cirrhosis, and other GI diseases. The outcome measures of fecal microbiome changes, varome changes, microbiome changes, or occurrence of any adverse event. So these are the stages of FMT. I'm not going to go through it. We have had the keynote speaker talking about it. And the operational definition for FMT responder was that post-FMT stool for culture done at 4 weeks is negative for MDRO. The second thing was to test for AMR gene depletion, whether AMR gene decreases with the help of PCR, what is the reduction in log. Now this is very interesting data. 429 patients rectal swab was sent. 80% of them in the hospital had MDR organisms. That is a very high figure. 71 patients satisfied the inclusion-exclusion criteria and were enrolled in the study. These were randomized into FMT arm, 38 patients, sham arm, 33 patients. The main organisms were E. coli, Klebsiella pneumonia, and Enterococcus faecium. Baseline characteristics, they were both similar in both arms, essentially to say that there were around 30 patients with acute pancreatitis, 30 patients with cirrhosis with decompensation, and these are the results. So if you look at results, they sound very encouraging. It shows that the outcome was proportion of patients with MDR organism colonization on culture, initially 100% because that was the inclusion criteria. After FMT, dropped down to 68.4%, very significant. Number of AMR genes detected by PCR also dropped down very significantly. So should we be happy with this result? Well, I don't know, but we weren't, because obviously this is not an uncontrolled study. This is a controlled study. In the sham arm, when we looked at the results, we found that, again, inclusion criteria was MDR organism, so 100% of them had the MDR organism. After intervention, 63%, again significant. Similar kind of spontaneous decolonization occurred in 4 weeks. Number of AMR genes did not decrease so significantly in that arm. Then we looked at another variable, that is proportion of patients with MDR organism decolonization on culture, or AMR gene depletion. In all patients, there was a trend, in the sense that there was a drop from 76 to 57%, around 18% delta, definite trend, but not statistically significant. In pancreatitis patient, it was statistically significant, whereas in cirrhotics, we did not find the same kind of signal. So again, there is some, what I would say, kind of trend out there. Good thing was when it came to adverse events, these are all very sick patients, and there were no major adverse events which occurred with FMT. So there was a trend towards intestinal decolonization, MDR and AMR gene depletion, and it was significant in patients with acute pancreatitis. Now comes the next thing. As they say, like behind every job that there's a successful man, there's a woman. Like out here for FMT studies, it is always the microbiome, and no longer it is a microbiome. You have to do the microbiome and also the varome. So in this study, all three were done: microbiome, and varome. 13 donors, 33 patients who underwent sham pre and post, 38 patients who underwent FMT pre and post. The diversity of the gut microbiome exhibited a significant increase 4 weeks post FMT. Alpha diversity was not so significant, beta diversity was definitely significant. These are the top 30 most prevalent species. These have been identified because in the sense they help tomorrow that if you are going to make a bacterial consortium and you want to look at which other bacteria you would want to build up in that. Gut microbiome also exhibited a significant increase in terms of diversity, but again, beta diversity 4 weeks following FMT as compared to sham treatment. And these are the top species of at genus as well as the species level, the microbiome which change following FMT which was given, again helping in identify what kind of fungal engraftment occurs and what are the species which are important. Then we also identified the phages which are the most important part out here, because you are expecting that bacterial resistance will be taken care of with phage addition or diversity. And this identifies the top phages which were increased in patients who were given FMT as compared to sham therapy. Donor engraftment, very important. Higher donor engraftment was associated with MDR or intestinal decolonization. Donor engraftment was however variable. Donor engraftment was seen much more in patients with acute pancreatitis and patients with chronic liver disease. So where does it all end up us with? It ends up with this, I think, this kind of picture where you're on the edge. The leap of faith, you would say, has been taken by one or two, not by everyone, because you are getting a trend when we're talking about MDRO decolonization or AMR gene, and at the same time, you're getting a very good signal at bacteriome, microbiome, virome levels. What could be the possible reasons that the results could not have been better? We are thinking perhaps we should have given a repeated FMT. We should have given a bowel wash for mechanical decolonization. We could have looked at choice of donor in terms of pairing of microbiota of donor and recipient. Then whenever we are giving FMT, essentially we are not able to provide for anaerobic bacteria. So if you're not providing for anaerobic bacteria, possibly the colonization resistance resides within the oxygen-sensitive bacteria, and that could be the reason that yes, you had a trend, you had a delta of 20% patients responding, but not that much as you would want. But nevertheless, science is by definition primarily about the hurdles. If there's a clear highway how to proceed, that is likely not a scientific route. And so I would end by concluding it is work in progress, but at the same time, a very optimistic work in progress. As I've shown you with the largest RCT till date, again, a well-conducted RC in terms of being double-blind, sham-controlled, and looking at all kinds of bacterium data. There's a trend towards intestinal decolonization of MDRO and AMR gene depletion. It is not statistically significant, whereby you can say clearly yes. In pancreatitis, yes, you're getting a much more stronger signal. There's a favorable microbiome and microbiome change. Hence, it has the potential to be a solution for AMR. Thank you so much for your attention.
Thank you very much, Dr. Vineet, and I must congratulate you for that excellent study. And in fact, we need many more of these to be certain on the impact of FMT on AMR. So we are very much interested, as you said. I mean, diarrhea remains very rampant in our population because of immunosuppressions. Almost 30% of our patients will suffer from whether it is because of change in gut microbiome or because of purely side effects of the drug. So we still want to do these studies, and I think one of these studies is already being undertaken. So with that, I think we possibly can take a couple of questions since we are running behind schedule. Yes, please.
Yes, sir. Thank you very much, sir. I am Nipun from Department of Hepatology, PGI. Excellent presentation and the study, sir. That concurs with our data as well. We did around 150 patients of colonization. 70% were colonized at admission. So the point here is...
so PGI and AIMS are equally good.
Equally
or
bad.
Not bad. I won't use the term bad. I said they're equally good.
And the point here was that after 7 days, this colonization further increased to 80%, and this was correlating with the infections, and the same bugs was present in the GI tract. That's besides the point. The point here is, sir, in patients with CLD, you didn't find any, engraftment as well as decolonization. One of the points which I was thinking is patients must be infected with, the MDR infections at the same time. So if the source control is inadequate, the engraftment rate is going to be low, I guess. And if we are doing in a cirrhotic patient, long-standing cirrhosis, if presenting to a different severity, different stages of cirrhosis, they are likely to have, more severe gut dysbiosis that is unlikely to be reversed with one fecal transplant. So I think, what is your opinion on recurrent transfusion, FMTs, and how should one ascertain the engraftment, the predictors of engraftment?
So in answer to your responses, number one, that we had excluded CTP more than 9 because we did not want very sick patients to come in, because this is one of the first studies and we didn't want to take a chance. Number two, FMT was done when none of these patients were off antibiotics. So MDR was established when they were discharged from the hospital, reconfirmed subsequently within 4 weeks before doing FMT. At that time, they were not on antibiotics. So now engraftment is something which is a very large question, because engraftment we have to look for strains rather than phenotypes or species. Number two, engraftment depends upon the factors in terms of if the donor microbiome and the recipient microbiome are almost similar, then engraftment will not occur. There has to be a significant difference in both of them for engraftment to occur. Secondly, I think this is a gray zone. We are not very clear what are the other factors which will decide engraftment. Repeated FMT should have helped. That is what we feel now. But when we did the trial, one was afraid to do 2 sessions of FMT in a patient who has just come out of the ICU.
I think we need more refined methods to do FMT rather than to do a colonoscopy. Yes, last question, please.
Yeah, so my question is, there are patients who had shown improvement, right? So did you see any donor-specific effect, like a super donor?
So we looked at, I've not shown the data, clinical responders versus non-responders. And also clearly the patients who had shown improvement, they had better donor engraftment. Donor engraftment was much higher in pancreatitis patients as compared to cirrhotics. Now this actually opens up a very good window this is for the people who are in pancreatitis field, like Group Jyoti is there. You have patients of acute pancreatitis of a mortality rate of 30 to 50% severe acute pancreatitis. And the mortality, the main cause of mortality is infection. So if 1 or 2 sessions of FMT at that stage can change the mortality rate, this is initial data for that. This is initial proof of concept for that.
Thank you.
Thank you very much, Dr. Vineet, for an excellent talk. And with that, we move to the